Case Western Reserve University's Institute for the Science of Origins proudly presents the Origins Science Scholars Program. The Institute advances the scientific understanding and application of origins and evolution of human and natural systems. The Origins Science Scholars Lectures are presented with the assistance of Case Western Reserve University's Siegel Lifelong Learning Program, College of Arts and Sciences, and Media Vision. Tonight, it is my pleasure to introduce Professor Michael Lewicki, who received his PhD from California Institute of Technology. Prior to his current appointment at Case in the Electrical Engineering and Computer Science Department, he was a fellow at the Institute for Advanced Study in Berlin, Germany. His research focuses on understanding neural information processing and computational principles in sensory encoding, perception, and scene analysis. Please join me in welcoming Professor Lewicki.
Tonight I'm going to talk about the brain and how we approach studying the brain and perception. So when we look at a complex scene like this, our impression is that we see it immediately. And what's always been on the back of my mind is how do we look at something like that and from the picture arrive at the understanding of the scene. And if you think about where the information starts, it's at the back of your eye, the retina. And the photoreceptors are at the very back, so the light hits the photoreceptors. They transduce those signals into electrical signals. And if you look at the photoreceptor array, they're packed very densely, right? So that's our connection to the visual world. And you can get a picture of what they look like by coloring them according to the color that they sense. And that's what you start with, all right? So what's always fascinated me is how do you start with this, which is essentially like a camera, right? It's just recording the intensities at different positions, but put together an understanding of what's out in the real world. And that's the problem animals solve every day, all the time. And I've been studying it my whole career. So to kind of get a sense of what the nature of the problem is, we can look at one of those photoreceptors. And initially it's just sensing light at a very specific location as you move your eyes around the room. And you can't really tell what the scene is, right? It doesn't make any sense. But if we back up a little bit, we have a few more photoreceptors and we get a slightly bigger picture of what the scene is. And at this point you can start to see features and maybe put them together and make conjectures about what might be out in the real world, but still it's very unclear. If we back out a little more, now we have a bigger picture. And now we can start to see features like edges and textures and things like that. But I'd be surprised if any of us can look at this and really put together the whole picture, right? Because there's just not enough information. Yet this is the problem your visual system faces. How does it start with all of this local information, process it, perform computations, and arrive at an understanding of the whole scene? So if we take all of these image patches and put them in the place where they are in the scene, what you should see when you look at this is just the immediacy of your perception. Right? So we fill in the whole scene. It's a complex scene, right? But you don't have any trouble understanding. You know it immediately. You can extract all kinds of information, like you know that it's a river, it's a stream going down rocks, you know that there are rocks and trees laying across. And it's all effortless, right? All subconscious. We have no idea really how we accomplish this. And part of the problem that we'll work toward is if you take some of these images, something as simple as an edge that we see, if you look at it, if you look at the details of the picture, all of those edges are contours to us. We treat them as similar, but the pictures, the individual patterns that you see are all very different. So at some level, we have to put together all these disparate images and arrive at some more general abstract concept. So in my own history, I started off really when I was in high school, thinking about optical character recognition, trying to get computers to recognize letters. I thought that would be a good project. And it was instructive because it turned out to be surprisingly difficult, as any endeavor in artificial intelligence or computer vision is. And I went to Carnegie Mellon because that was kind of the center of artificial intelligence and robotics. And I remember a very first experience seeing a robot, and it looked kind of like this. They had a robot on the sidewalk, and I thought, wow, that's a robot. You know, this was in the early 1980s. And I kind of approached it cautiously, and I was looking at it, and I was looking at the camera, and it was kind of, every once in a while it would move a little bit. And I thought, oh, that's really neat. But then when I kind of rounded the corner, I saw there was this orange tape going down the middle of the sidewalk. I said, well, what's the orange tape for? Well, that's how it follows the sidewalk, because it needs the orange tape to interpret the world. I thought, there must be a long way to go. So to give you a sense of how different this is, so that's kind of the computer vision / robotics approach, right? You treat it as an engineering problem. But the engineering problem is also incredibly difficult. So here's a different way to look at an image. So these are two images, but what Kirsten and Yuli here have done is represent the pixel intensity at a given point by a height on a map. So you see it as like a three-dimensional surface, but it's the same information. But now we've taken your visual system out of the equation, right? You can't interpret this because it's not in a format that your visual system is used to seeing. So look at that and see if you can figure out anything about that image. That's the problem your brain is solving. And I've given this demo for years, and no one has ever gotten it. So look at it. Look at all those features. You know, they're all little lines, little textures, and so on. But now I'm going to show you another picture, and be aware of the immediacy of your perception. And it's also, again, of a complex scene. It's a bicycle leaning against a tree, and you can see the grass in the background, you can see the sunlight patches, all kinds of information, right? So now that you know that, what's the other image? Well, it's also a bicycle, and it's the same bicycle. So that's the problem your visual system solves. It takes this low-level information and somehow puts it together, and you can immediately recognize very general things and understand very general things about the scene. So around the same time, when I was an undergraduate at Carnegie Mellon, I also got very interested in the brain. It was sort of the rebirth of neural networks around that time. There's a lot of excitement about the possibility that we could make computational models of how the brain worked. Computers were becoming more powerful. We had models of how neural circuits worked. And there was a lot of excitement about how you could go about understanding the brain and what it did from a computational point of view, meaning you could make models of how it processed it. And if you looked at neuroscience around then, and really still to a large extent today, you could ask, well, what does the retina do? And if you asked a retinal physiologist, they would say, well, we study cells in the retina and how they interpret the world. The retina sees the world in terms of little spots of light. And they're little spots surrounded by kind of a shadow, a darker area. And the term for this is center surround features. And that's the kind of feature that the retina detects about the world. And so it kind of makes sense that we start off this way. If you go deeper in the brain, so if we go back to the visual cortex, that's where the retina sends all the information via the LGN, then we can again ask the question, well, what do the cells respond to there? What are the features that they see? And the answer to that question is largely edge detectors. They see lines, dark next to light lines. And so it's very good at detecting boundaries in the visual scene. And that made sense too. Like, we see the world, and one of the first things the brain does is start to put together edges. Well, this inspired me to go off and do a PhD in neuroscience. So I went to Caltech because they had just started this program in computation and neural systems. And so you learned physics, and you learned electrical engineering, and you learned computer science, and you learned biology. And the hope was that you put all those things together, and you'll be able to make theories of the brain. And so I was very fascinated by acoustic representations. And I was in the laboratory of Mark Konishi. And in the back of my mind was always this quote by David Marr, which was that, because the whole thinking of computation is what's the algorithm that the brain is using? And David Marr was the founder of the field of computer vision. And so he basically defined many of the major problems that are still studied. And he also was a neuroscientist. He made some of the first computational models of brain areas. And what he said at the beginning of one of his books is that an algorithm, or the computation, is more likely to be understood by understanding the nature of the problem being solved than by examining the properties of the system. So when you're a neuroscientist, all you can do is examine the properties of the system. It's very difficult to think about what the system as a whole is doing. And so I spent many years trying to get data. So it's technically very difficult to record from neurons, and even more difficult to record from them intracellularly so that you can see the voltage and what's going on inside. And so this trace here is a neuron. It's in the songbird, and it represents something about the bird song. And it has a very interesting response. The pattern below is the waveform of the bird song. And this cell responds in very specific ways to the bird song. Right? Those are overlaid traces to different presentations of that song. And you look at that and you are just fascinated because it's so regular and it seems to contain so much information. And so you report that, but then you're left with the feeling that you don't really understand how the whole system works. How is it putting all this together? You have all these little pieces, but you can't see the big picture. So after that, I was more motivated to go into computational research. And what I was interested in is, are there principles that the brain follows? You know, if you're trying to describe the system, you need to have some framework. And what were the information processing principles? I mean, partly we were trained in physics, and physics is fantastic because it describes the physical world, and there's all these physical laws, and everything's very neat, at least if you don't know too much. And in the brain, nothing is neat. So the inspiration of why it might possibly work comes from eyes. And if you look at eyes, there are millions of eyes, and they're all different, and it seems like they're just kind of random. But they're not random if you understand the principles of optics. And so this is one thing where physics really works, because if you understand physics and how light propagates through refractive media, then things start to make sense. And what you realize is that there's only a few types of eyes. There are things simple that basically make a shadow, but then there are also lens-type eyes that work by refraction. There are eyes that work by reflection. And you can describe all these different eyes and how they process the light. And it even works for very different eyes, like compound eyes in insects. So the physics works. There are underlying principles that make sense of all this variety. And you can even explain how a particular eye is adapted for what the animal has to do. So could there be things like this, but for neural information processing? And there's even more hope because people were simulating the evolution of the eye. So you can introduce like a few percent variations, say in an array of photoreceptors and their physical structure. So you can evolve that in maybe a few 100,000 generations. And each generation is a little bit better than the rest. And if you also allow the refractive media to vary, then you can evolve a lens. And each time, the thing that it's doing, the advantage that it's giving to the animal gets a little bit better. So that's very interesting. Evolution kind of is evolving according to underlying principles, constraints of the physical world. So when we look at the retina and other parts of the brain, we can ask, well, what problem does the retina solve? Because when we take the light, we can explain how the light gets projected onto the back of the retina. And maybe that took hundreds of millions of years of evolution to evolve. However, all that time, the retinal circuitry is evolving too. But what was it constrained by? What problems was it solving? So that's the question. And at this point, I will take questions from the audience.
If you're perceiving by edge analysis, as you discussed before, how do you perceive color? And because there's no edge to color.
So color is a whole different story. And edges is really just one of many things that you could talk about, even starting at the retina. And you could also talk about motion, which is another very salient feature that animals pull out. So the whole field of study is about trying to identify just those kinds of questions. How do we perceive different kinds of features in the world? And edges is just kind of one aspect of it, a tiny fraction.
how much does experience come into play? You know, like when I look at that scene there, I immediately know it's a wooded scene with a tree trunk, etc. But that's because I've been in woods and I've seen these things many, many times.
That's a very good question, because when we go deeper into perception, one thing early on that you realize is that it's mostly inference. And you have to deduce what's there, because what you're starting with is very little information, and that information is noisy. And so your experience plays a big part, a crucial part, in allowing you to interpret what would otherwise be ambiguous. So a good example of this is speech. You can listen to speech in a very crowded, noisy room, but you can still understand it, even though acoustically many of those things are missing. And it's the same with like a scene like this, the woods. If an animal is behind the tree, it's partly missing, right? But you fill it in because you have knowledge of what must be behind the tree according to the structure. So that knowledge really is present at all levels in the visual system.
You mentioned motion, and I thought, have you done comparative analyses of the eye structures of something, of the eyes of a bat that's trying to catch a flying insect at night versus the eyes of a camel that's trying to find a water hole in the desert in the middle of the day?
Yeah, that's an excellent question. So that motivates many kind of evolutionary biologists or physiologists or animal behaviorists. So there are a multitude of fascinating stories that go after exactly those kinds of questions. How have animals evolved? How are they adapted to their particular environmental niches? And there's stories at the behavioral level, there's stories at the perceptual level, there's stories at the sensory level. And you know, just to go back to your bat example, bats hunt with echolocation, right? And depending whether bat is above the tree level or below the tree level, it has a very different acoustic background. And so bats have evolved different echolocation signals so that they work best in those environments.
Thank you for joining us. You've been watching Dr. Michael Lewicki discussing the information processing circuitry that transforms what our eye detects into what we see. For more information on the Origin Science Scholars Program, please visit the Institute's website at origins.case.edu. In the next part of the talk, Dr. Lewicki will discuss the model he developed for how the brain processes visual data. Now, back to the talk.
The question we had is, what problem does the retina solve? Because if we understood that, we could ask the question, what's ideal? So another dilemma you're faced with when you're doing theoretical work is, do you try and model the system, or are you trying to describe all the details and work closely to match what's actually there, what you observe? Or do you take a more top-down approach and try and describe the computations and whatever other important properties of the system there are from a very abstract point of view? So when we look at something like the retina is incredibly complex, and there are at least 20 varieties of cells in the retina and many different kind of subsystems that the retina have. And we can't hope really to describe them all. So to kind of get a toehold, what we want to do is look at the retina, understand what goes in, what comes out, and try and make the simplest model we can of that process. So go for maximum abstraction, maximum informative abstraction. So like we said before, the signal starts with photoreceptors transducing the signal, and then there's the other cells in the retina that transform the signal into a code that's transmitted by the retinal ganglion cells that goes down the optic nerve and to the brain. And what we can do is we can measure those signals, and that's how they figured out that cells are center surround. And so this way allows you to characterize how a neuron behaves. And so if you know the input and you know the output, you can ask the question, well, what's a best model for characterizing that response? And then furthermore, if you know how the whole population behaves and how it's organized, then you can try and think of a theory that will explain why it is the way it is. So first let's look at what a center surround feature is. So we have the photoreceptor array there, and overlaid on it is the center surround thing. And what this means is that if you take a spot of light and shine it on the middle, which is the excitatory subfield, then what happens is that cell responds. And the way a cell responds is it generates what are called action potentials, spikes. And so it's a little point of electricity that it sends down the axon, and that's its signal that goes to the brain. If you shine that same spot of light on the surround, which is an inhibitory subfield, then the background activity goes off. And so the cell is inhibited by light in the surround. And so that's why this is called an excitatory center, an inhibitory surround, and a center surround cell. So when we set out to make a model of this, we knew that we could derive efficient codes. And what we started with is just the image, just as a pixel array. And then we describe how the image is projected onto the retina. So that involves blur. And then it hits the photoreceptors, and the photoreceptors transduce it to a signal. But they can only do it at so much precision, and that involves noise. So we model that too. That's an essential aspect. So we're trying to be as simple as possible, but no simpler. Then we have the encoding, something that actually generates the code. How do we take the pattern that appears at the photoreceptors and transform that to a more efficient code that gets sent to the brain by the ganglion cells? And so here's the evolutionary hypothesis, that the problem that the retina is solving, or one of the problems, is that it has to encode the visual signal in a way that's maximally efficient, that transmits the maximum amount of information to the brain. So if we view that transformation as kind of an aggregation of all those photoreceptor response, we can ask, well, what's the optimal way to aggregate those signals into something much more compact, into an efficient neural code? So we do the retinal encoding, and that results in retinal cells, retinal ganglion cells, that gives you a retinal code. And what we can do is compare that code to what we actually measure physiologically. So this is now just a computational problem. We can set an objective function, so a goal for the system, which is to minimize the decoding error. So just like when you take a picture with your camera, it stores that image as a JPEG. And if you want it very compact, it's going to lose information, and you might see artifacts and things like that. If you want it to be encoded very exactly, well, then the code is going to be larger, but you're going to have a more accurate representation of that image. So the retinal code is the same way. But the retina has to work with cells, and so those cells are very noisy, so it doesn't have much to work with. But we can tell how good it is because we can decode the image, and then we can compare that decoded version to the original version and ask, well, how do we make that better? And by that process, we can optimize the weights and predict what would be theoretically optimal. So we also have to add some additional constraints, biological architectural features or biological costs. And so one of them is diversity. So at the fovea, the cells are very dense, and there's kind of a lot of resources. In the periphery, it's much more sparse, and so you don't have as many cells to code the information. And so that should predict different codes. We also had the constraint that I mentioned, which is that cells can only encode about 1 bit per spike, which is very low. Right? You're not talking about a floating point number here. And then there are resource costs. When a cell sends a signal, that incurs an energy cost. And when you have a connection to other signals, there's kind of a physical cost in the amount of space that it takes up. So we put all those into the mix, and what we do is we derive the optimal code. Okay, so I'll get to that in a minute, but let's first look at how we might compare what are traditionally considered optimal codes. And so engineers have worked on coding for a long time, and there's a lot known about this. So if we take those same weights that we're using to do the transformation, we can put them through this retinal model and ask, well, how good does an optimal code go? But the answer turns out to be terrible, because it's not taking into account the noise. And it's actually 34% error. And we can't really see like this. So let's look at what's happening. So we have our model. The weights are set according to these engineering codes, mathematically optimal. But the problem is they assume arbitrary precision, right? Which isn't what the retina has. And they're essentially an array of numbers, right? So that loses a lot of information when they're subject to noise. So what we want to do is optimize the code that's robust to that imprecision, that's robust to the noise present in the system. So the theory optimally accounts for that. And it assumes that the neurons have noise, and it figures out what's the best code that will give the best reconstruction and therefore transmit the maximum amount of information to the brain. So then the information is retained. And so it's important to understand that this is the same architecture. It's the same number of cells, It's the same number of weights. All we've done is change the values, so they're optimized to a different objective function. So now when we decode the images for this code, how does it do? Well, it does much better. We're down to 4% error, and that's just with 1 bit of information per unit. So that's very encouraging. But now we have a theory, and we can say, well, what are the features that it's using to encode these images? Do they at all look similar to the retina, or is it just some other, another type of engineering code? Well, to do this, we have to look at the weights and look at the pattern of connections between the retinal cells and the photoreceptors. And that's a 2-dimensional pattern. And initially, it's just random because it's unorganized. We optimize it, right? So what we want to do is look at those patterns and ask, how does it change for different objectives? So what we're expecting is the center surround architecture. And when we have no constraints at all, somewhat surprisingly, there's no pattern. It still transmits information, but it kind of looks unstructured. Okay, what if we add some resource cost? Well, if there's a neural activity cost, so there's a cost to coding the information, then we start to get structured receptive fields. And they're like the edge detectors that we saw before. They respond to adjacent regions of contrast, a light line next to a dark line. But that's still not what you see in the retina. And so this is where the resource constraints become important. And when we add the constraint of weight locality, that things should be as compact as possible, take up as little space as possible. Then we see center surround architecture. So if we expand one of those, we also see that when you look at the 2D receptive field and look at how they're organized in the retinal population, it matches what you see physiologically. And then if you go out to the periphery and say, how does the code change? It also matches what you see physiologically. So this gives us a new way to interpret what it's doing. The retina is forming a robust code to transmit the maximum amount of information to the brain subject to these resource constraints. Okay. So what's beyond coding? So that's one kind of approach to understanding the brain, to understand what problems it solves. At the very start of the brain, your connection to the physical world, it has to code the information, and it makes sense that that should be done efficiently. But what about higher levels? Well, if we go to the visual cortex, we said there are edge detectors. And that's true. The features are like edge detectors. But you can only get so far using these simple features. So let me illustrate that. So the problem is that simple features can't detect complex edges. And so if you look at this raccoon, you have no trouble seeing it, right? And the edge detectors work when there's a sharp, bright, dark boundary. Okay, so what I'm going to do is, and this was work by a postdoc of mine, Chris DiMattina, and you scan the visual field with a bunch of edge detectors, and if one of them responds, you put a little point there. Okay, so we'll see all the places in the visual scene where there's an edge, where it thinks that there's an edge. So are you ready? And it turns out there's a lot of edges, right? And so the dark ones that are next to the light ones show up. It detects those, but not much of the raccoon is there because a lot of those edges are really texture edges. So we can also illustrate a different problem, which is related. When we look at a complex scene, one of the first problems you have is how do you organize all of this information? You have all of these features. But the problem you face when you're trying to understand the scene is to put that together into a more abstract representation, to recover information from the external scene so that you can act on it, right? So that you can navigate the scenes, you can recognize where the prey is, all those things. So the problem is that you look at the scene and the bark on the tree, for example, those are all different textures, but you recognize it as being part of the same surface. If you look down with the red contours, that's a tree, and you see that as a single contour, but all those little image patches are different, right? They're different images. So this is a generalization problem. This is why we called it perceptual generalization. And so this is work with a student of mine, Jan Karklin. And what we're interested in is what's a problem, an essential computation that you would have to do early on in the brain. And we explored this idea of perceptual generalization, that somehow you had to go from all these different images to something more abstract, something that put them all together. And this is analogous to something we can think consciously about, like recognizing a dog, right? You've seen a dog that you've never seen before in your life, but you recognize it as a dog, right? Even though it's a unique image. So how are you making that generalization? How are you going from the specific to the general? And that same problem occurs at the very lowest level. How are you going from the specific patterns of features to the more abstract concepts like surfaces or contours or foreground or background? So I won't go into the details of the model, but I'll just kind of give you the gist, right? If you start with simple features, those are edge detectors. And if you look at different responses to, like from the scene we just saw, and you try and make sense of them, what you see is that, well, it's all jumbled together. So what this shows is the joint activation of two different features. And so the squares here are all over the place. The circles here, which correspond to the contours of the tree, are all over the place. And the same for the others. So it's all mixed up. There's no way you could look at the activity of those patterns and figure out that you're looking at tree bark, or figure out that you're looking at contour, where you can't generalize. As Patricia said, you can't see the forest for the trees. However, mathematically, we can describe this problem. It's really a problem of learning statistical distributions. If you can generalize to these distributions, then you can represent this same perceptual information, but at a more abstract level, in a way that puts together many different patterns. So this is a nonlinear computation. It's an algorithm, and it's kind of like putting a puzzle together. And when you look at those abstract features and look at how it responds to those same patterns, what it has to do is integrate all of those different features. So this is another plot showing the weights that integrate these local features across a visual field. So what this shows is that it's integrating from hundreds of simple features. And this is just one unit at this more abstract level. And it's sort of an aspect of this computation. So when we look at this abstract representation, we can ask the question, well, can it distinguish these different types of images? So this is an abstract feature space. And when we plot in that abstract feature space, lo and behold, everything makes sense. All of the contours in the tree edge are grouped together. They're part of the same perceptual space. The tree bark is similar, but they're all grouped together. And the same for the foreground and the background. So it solved this problem of perceptual generalization using these algorithms, right? This computation. And more remarkably, you can compare it to the physiology, and it even accounts for a lot of non-trivial physiological properties. So it's a theoretical or functional account of some of these mysterious properties. You can also illustrate how it interprets a scene by looking, say, at this scene. So if you group local features, what you find is that the colors correspond to different clusters, but they don't do a very good job of grouping different regions together. And if you do it in the abstract feature space, you get a very different picture. So now we'll do that same thing, but using the abstract features. And now the log is all part of the same, more abstract concept. And the most interesting thing is if you look at, if you look at this contour, this edge here is the same as the edges up here. So that perceptual dimension is abstract. It takes all of these different patterns, and at that level of the representation, it's the same. So I'll take questions.
So all of these pictures have a massive amount of information, yet we know...
Sorry about that.
When we look, no, but we know when we look at something, we don't necessarily need all of that, so people are very good at being able to fill in the spaces. And so the question I have is, how much do we fill in the spaces and ignore what the rest of the information is? And this is part of the problem with eyewitness accounts, because they've seen part of it and they've filled in the rest. and they haven't actually interpreted it, and so everybody has a different picture of what they're seeing, not because the picture's different, but because they've used different parts of the picture to fill in the hole.
That's a very astute observation. And like I mentioned before, that this idea of filling in from your prior knowledge or your experience is an essential part of perception. Because like you alluded to, there's just too much information to process it all, right? And evolutionarily, you would want to rely on accurate assumptions in the presence of little information, and that also allows you to act very quickly, right? The downside is that you can be wrong if your assumptions are wrong, or if the information you thought was missing and filled in actually wasn't there. And there's a lot of studies in perception about the ability or inability of people to fill in accurately. And one impressive one is something called change blindness. And if you slowly change a scene, and so you remove motion cues, what happens is that you can remove large things, like an entire person from a scene. Actually, this originated from a gorilla. So there's this famous experience where these people are passing a basketball around, and they're having to count how many times one team touched it. And so it's an attentionally demanding activity. And then in the midst of all this, a gorilla guy in the suit, walks across the stage, and nobody notices. Subjects watching this who are doing this intensive counting just don't see the gorilla, because their attention is focused on the information they need. And that probably goes on at a very deep level.
Your perception depends upon what you expect to see. And looking at that scene up there, I expect to see leaves and trees and ground and clutter, whatever. And I wouldn't expect to see, I would expect to see an animal, but I wouldn't expect to see a fish coming across. So that's where the scene is set. Mm-hmm. And you can experience it. If you're watching for a house number, say as you drive down the street, you're concentrating on that, and you don't see other things or the cars in the driveway or whatever.
That's right. Your attention has to be very selective, because there's just simply too much information in the world to process. And so that's kind of a fundamental aspect of perceiving complex things. The only way that's possible is if you build a representation of the world, and you're relying on that representation to be accurate. But that also means that it can be fooled. And so there are all kinds of illusions and things that you wouldn't think people would misperceive, yet they do.
How much of your work is being used by people like Tesla and Amazon in developing sensors for automated cars?
What's really hot right now in artificial intelligence is deep neural networks. And deep neural networks are being used to solve all kinds of problems, including self-driving cars. And so I mentioned that when I first started at Carnegie Mellon, neural networks were just re-emerging. And so this was like 30 years ago. And so for a long time, that research was just really abstract research. You wanted to process information in a way that was similar to the brain. And the features they focused on were things that are massively parallel and things that could learn structure from the data. And so this kind of research, the idea that you could have an optimal representation, the idea that you could have a complex model that would acquire information from the data, is now an essential aspect of training these very large models. Because without that kind of learning capability, it wouldn't be possible to train these large models. So they use these same kinds of features at the lowest level. And then what they do afterward is, instead of trying to mimic what the brain is doing, rather they use machine learning technologies to do classification and other types of processing. So it's part of really the overall effort to understand, I guess, information processing in perception.
We hope you've been enjoying the Origin Science Scholars Program with Dr. Michael Lewicki. Dr. Lewicki is Associate Professor of Electrical Engineering and Computer Science at Case Western Reserve University. In the second part of our talk, we learned how the brain decodes complex scenes. In our final segment, Dr. Lewicki will tell us how his ideas about processing of visual information carry over to our processing of auditory signals. Now, back to our talk.
So in the last part of the talk, I wanted to Well, first sum up what this is. So it's a general theoretical approach. And you start off, like we said, defining the problem. It's most important to ask what problem is the system trying to solve. And then you spend a lot of time trying to think of how would you solve that problem? What's the model? What's the algorithm? And so on. And then once you have that, now it's a mathematical problem. How do you optimize that to natural signals? Because you assume that the brain is optimized to the natural world. And then all of this great theory might just be wrong because it doesn't match the experimental data. And so you have to repeat, right? You refine the model, you improve the algorithm, and so on. But what I want to focus on for the last part is the generality of the approach, that we can take these exact same ideas, the same theoretical approach, and apply them to a completely different system, which is the auditory system. So the question is the auditory system adapted to natural sounds? So like vision, you start with a signal, but this time it's a time-varying signal. Well, vision is too, but we'll focus on the time now. And the auditory nerve is what sends the information to the brain. So your connection to the acoustic world is the signals coming down your auditory nerve. And we can measure those, the neural activity, and they're also in terms of spikes. And what physiologists can do is characterize how a particular auditory nerve responds to sound. And so we know that it responds selectively to, say, signals at particular frequencies. And if you look at the structure of those features, so this is a little acoustic feature, right? It's a little wavelet-like thing. And it has a very characteristic shape. So it has a frequency, which is described by that function. And you can see that in, it can be a high frequency feature, it can be a low frequency feature, so it's something that varies. And over the auditory nerve and the cochlea, it varies systematically from low frequencies to high frequencies. Now what also varies is the envelope, so the amount of time that it integrates this acoustic information. And so that describes what's either called the bandwidth or this gamma tone, this gamma shape. And what we want to know is, well, why does the auditory nerve have these particular shapes? So the first problem is, well, how do we make a model, right? We need some way to describe how we transform the acoustic signal into a representation, into a code that we can then optimize. So let me show you how we go about doing that. So we take some sound waveform, and then we look at those acoustic features, the gamma tones. And so each little blip here represents the acoustic feature at a particular point in the waveform. So here's a low frequency acoustic feature that represents this structure. This one is a high frequency acoustic feature that represents that structure, and so on. And so with that code, what you can do is recover the information. And this was work I did with Evan Smith, who is now Vivian Ming, back when I was at Carnegie Mellon. And what we were interested in is, well, what are the optimal codes? So the first problem was, how do we build something that can actually encode the information in an efficient way? And then once we had that, now we're interested in the problem of what's optimal. Okay, so the first discovery was that this type of representation is very efficient. So if you take a speech waveform, for example, and describe it in what we call a spike representation, a representation of the acoustic signal in terms of these small acoustic features. Well, the traditional representation is called a spectrogram. You plot the spectrum as it varies over time. And the way you compute this is you have a 30 millisecond window, you calculate the spectrum over that window, and then you slide it along. And so the problem is that it tends to blur the sharp acoustic features, right? You lose precise time information. But with this other representation, you get extremely precise time information because it's accurate to the sample. And if you look at the spikes here, those spikes represent the little blip. So this is the sound Vietnamese, and this little blip here is the T part, because it's the point in time when the tongue comes up and hits the roof of the mouth. And so it's very brief, and it's different from the underlying vowel. And that's all you have to know that that acoustic feature occurred. And in the spectrogram, you see that that signal is kind of blurred out over about 10 milliseconds and spread out over a lot of different frequencies. But in the spike code, that same feature is encoded by just 4 spikes. And those 4 spikes encode the T, and the rest of the residual pattern is the vowel. Right? So it's a really efficient representation. So now we can do some theory. Okay, we have the kind of computational architecture. And so this is the paradigm. We want to know, how did the auditory system adapt to the natural acoustic environment? Well, evolution has been working at this for a long time. And it evolved the cochlea, it evolved particular codes at the auditory nerve, and we know a lot about the properties, right? The properties of the system. So we know that they vary in frequency, and we know they vary in their integration window. And what we would like, ideally, is to predict what these features are from a purely theoretical point of view, meaning we have this idea that we should form an efficient code. We're going to derive the optimal features by only asking what's the best code for the natural sound environment. And so that optimal auditory code should predict the individual shapes, those features, but it should also predict how they're organized in the population. So what is it? Well, we don't know. It's a theoretical prediction. We hope it's the same. But a problem here is that what it predicts depends on what the data is. So first we have to answer the question, well, what are natural sounds? Oh, interesting question. So what we did is set out to collect a bunch of different sounds. And our hypothesis was that the auditory system has evolved really to either escape prey or find prey or do animal communication, right? Find a mate. And this involves sound localization. It involves animal vocalizations. And so we can divide them up into roughly two groups, vocalizations and environmental sounds. And then it turns out environmental sounds we needed to subdivide into transient environmental sounds and then ambient environmental sounds. So the vocalizations are things you're all familiar with. And so if the sound is on, that's a fox. All right? It's very harmonic. Here's a squirrel. You know, it's chirpy, but still very different from a natural sound. So if you were doing communication, those sounds would stand out from the background. But if you're doing sound localization, well, then you're interested in animals moving. And so if you're in a forest setting and you walk on leaves, it sounds like this. It's a very different type of sound. It's transient, right? Very brief, very sharp. If it, cracking twigs and cracking branches, also the same way. And then there is ambient sounds. So these are very brief, but ambient sounds have a longer duration. Rustling type sounds, or stream type sounds. So what we did, we threw all those into the mix, and we said, we're going to start from a random set of acoustic features, and then use our information theory to derive the optimal set of acoustic features. And here you can see evolution in action, at least in simulation. And so the plot will show the signal-to-noise ratio, which is the fidelity of the signal, in blue, and then in red will be the code length, and the code length will go down over time. And what you'll see is that each of these random patterns is one member of this set of acoustic features. And so there it goes. And they very quickly evolved to things that look very much like the gamma tone functions. And remarkably, if you compare these functions to the auditory nerve, so we can take these features that we learned, and then physiologists, this is from Laurel Carney, have large databases of acoustic nerve response properties. They're called RevCore functions, but they're kind of like the acoustic feature that predicts how the auditory nerve will send the signal to the brain. And so we take that first one, we see that, well, there's an acoustic or an auditory nerve filter that matches exactly. Take the second one, well, there's an even better match. And in fact, for every theoretical feature that we learned, there's an almost exact correspondence of something that was measured at the auditory nerve. And so remember, this started from random. So it's if you take this random set of functions and just ask the question, okay, take these sounds, derive the optimal representation. It can be any shape you want, it can be any length. And they just happen to correspond to what you measure at the auditory nerve. So that was very nice. And what we found is that you needed to divide the acoustic space into these general groups. Animal vocalizations was roughly 1/3. Transient environmental sounds was about another third. And then ambient environmental sounds was the third, and these were the exact ratios that we wound up getting the best fit to the data. But this story started, this is the only time this has happened in my scientific career. And when Evan, now Vivian, we were talking about this project, we said, oh yeah, this idea should work, and let's go try it. And Evan worked it up, and in about 2 weeks had a result. And so I walked in, looked at the results, and they were amazing, because they were exactly the auditory nerve filters. But we hadn't yet gone out and recorded all those sounds. What we did is just took data we had lying around in the lab, which is speech. So if you look at the auditory nerve population, so this is the center frequency versus the bandwidth, right? And so this gives you a picture of how the auditory nerve population is organized. And what you see is that in general, the integration time decreases, or the bandwidth increases, as the center frequency increases. So if you just take environmental sounds and plot them in that same space, they don't match. And they don't look like the auditory nerve filters either. If you take animal vocalizations, they don't match either. And so this is why we were scratching our head, right? What's the right mix? And so Evan realized that you had to subdivide the environmental sounds into transient sounds and ambient, sustained sounds. And when we did that, natural sounds, we called it, it matches pretty well. But that took a year of working, going out in the field, collecting all these sounds. What we had in 2 weeks was the speech. And the speech almost fits even better. So that was really kind of puzzling, because another thing that I didn't tell you is where this acoustic data comes from, right? If you take natural sounds, do this optimization, you get auditory nerve filters. If you take speech and do this optimization, you get auditory nerve filters. But where did the auditory nerve filters come from? Well, they were recorded from cats. So how is that possible? Are cats evolved to human speech? Probably not. So it turns out speech very closely matches the natural composition of sounds. Vocalizations correspond to vowels, transient sounds correspond to hard consonants, and then ambient sounds correspond to soft consonants. And it just so happens speech sits right in the middle. So more concise explanation is, well, cats aren't adapted to speech, it's the other way around. It's more that speech is adapted to the pre-existing mammalian auditory system. Because if you already have a system that's good at doing animal vocalizations, communication, it's good at doing sound localizations, it has to process all these sounds. And normal animals want their vocalizations to stand out from the background noise. But if you're face-to-face and you want to really communicate a lot of information, well, there's a lot more information you can send by having these other types of sounds. So it makes sense that you would evolve ways to produce those sounds. All right, so with that, I will acknowledge all the people who contributed to this work over the many years. So Ivor O'Doy was a postdoc at Carnegie Mellon and here at Case, and he was largely responsible for the retinal coding work. Many people looked at higher order features, including Jan Karklin, Chris Dimachina, and Zhujing Chen. And then Evan Smith, now Vivian Ming, did all the work on auditory coding. And then also thanks to all the grant support I've had over the years from NSF, ONR, and DONE. So I guess now is time to break for dinner. Thank you.
The Origin Science Scholars Lectures are presented by Case Western Reserve University's Institute for the Science of Origins, with the assistance of the Siegel Lifelong Learning Program, the College of Arts and Sciences, and MediaVision. For more information on the Origin Science Scholars Program, including a full video archive, please visit the Institute's website at origins.case.edu.